Welcome to everyone. Thank you for attending and joining us today. So this is the second lecture in the TPSF, the Trevor Platt Science Foundation course. As I said last time, the lecture from Dr. Lauren Bierman, it's great to see such a high level of interest. We've got attendees from all over the globe, as well as speakers from all over the globe. It's really great to see. So I think it's best if I introduce the speaker and we crack on now. So the speaker today is Dr. Anna Dogliotti. Following her PhD, Dr. Dogliotti went on to work for the Royal Belgian Institute of Natural Sciences and the Institute of Astronomy and Space Physics with the Universidad de Buenos Aires. Currently works for the National Scientific and Technical Research Council, performing research in a range of remote sensing and oceanographic subfields. Her main activities are related to the validation of satellite products using data collected with radiometers, the evaluation of atmospheric correction algorithms, and the development of biooptical algorithms to evaluate and improve satellite-derived products in the Argentine seas. Especially with some work on the very complex turbid waters of the Rio de la Plata. So we are in very good hands for a discussion on today's topic. So it's my pleasure to hand you over to Dr. Anna Dogliotti for the second lecture of the course, which is an introduction to ocean color. Thank you.
Thank you, Thomas. I will share my screen. Okay. So thank you very much again for the introduction, and also thank you for inviting me. As a former trainee of Trevor Platt, I've been part of his courses in the past, so I'm really honored to be here and giving this talk. I hope I will contribute to this course. So I will talk, as Thomas said, about the introduction in ocean color. In the previous talk, yes. So Lauren, she's been talking about, in general, about remote sensing. And she mentioned, of course, that it's the best option for gathering synoptic measurements with sufficient temporal and spatial scale for marine ecosystem monitoring. And she mentioned about sea surface height, sea surface temperature, and ocean color. So in this lecture today, I will focus on the fundamentals and optics related to ocean color. So I will try to go from the more from the basics to the more complicated things, but it will be hopefully a good. So I will start with what's ocean color, or what we call ocean color remote sensing. And actually, the goal of using remote sensing of ocean color is to derive quantitative information on the type and concentration of the substances present in the water. And this can be retrieved from variations in the spectral form and magnitude of what we call the ocean color signal. I'll go more in detail about this, but this is mainly the definition. So basically what we see is that the water can have different colors. This probably you have been seeing that. We can see very blue, very brown, and green waters. So basically the question here is, what is the color of the water and what does this depend on? So going to the first question, so what's the color of the water? So basically what we perceive of the color depends on the intensity of the light that is reflected by the water body at different wavelengths in the visible region of the electromagnetic field spectrum. So basically what we have the white light, which is the light that comes from the sun. The visible part of the spectrum is between what we call between 400 and 700 nanometers. So that's the visible part. So that's what we see. So if we put the blue water, so the intensity of the light coming from, this is just an example now, it's coming from the water, which is blue, it will have a peak around the 400, which is the blue part of the spectrum. While if the water is more greenish, you will have a peak. This is theoretical, I mean, it's not a true spectra, but will have a peak or more intense reflectance in the green part of the spectrum. So, and now going to what the color depends on, why the color changes. So basically, the color will result from the change in the type of energy, the radiation that comes from the water after interacting with the substance present in the water. So basically, it will depend on what is in the water. So going a bit of what can happen to light when enters a body, interacts with material, let's say. So basically, light can be either reflected, so it doesn't really interact with the water body or the body, just specular reflected. It can be transmitted. It passes from one medium to another. It can be scattered, which means the change in the direction of the light, and also can be absorbed. So it is transformed. And you can see another way of looking at it in the right. You can see from the incident light interacting with a particle. So you can see the change of direction, which is all the scatter in arrows. And also we can divide it in the what we call back scatter light. So what is scattered in the backward direction, so where the incident light comes from, and also in the forward scatter light or forward scattering, which is the part of the light that is scattered in the direction opposite to where the light comes from. And also the light can be absorbed. And then we get what is unscattered light that goes through the particle. So going specifically into the water, what happens? So we have incident light that comes from the sun, what we can call downwelling irradiance, type of radiance, energy basically. Part of it can be reflected. So reflected, in this case, what we call sunglint, which is a specular reflection. It's like when we look at the water and you see this, it's like looking at the mirror. You see this mirror, you see it's actually the light that is specularly reflected. So it doesn't interact with the water, but only with the surface. Another part can be transmitted, so passes the surface and gets into the water. Inside the water, as I mentioned, it can be absorbed and scattered by the different substances present in the water. Part of the scattering I mentioned can be mentioned like backscattering. Part will be backscattering in the direction of the incoming light. Fluorescence is another type of scattering. Inelastic, which change not the direction, but the wavelength of the scattered light. And so finally, after all these interactions, part of the light will emerge from the surface, what we call water leaving radiance. So basically, what we usually talk about, and when I mention what is color, I talk about reflectance. And actually, reflectance is the relation exactly between what came, what arrived to the surface, and finally what was leaving. So it's the amount of energy that is actually reflected. So put it in with the nomenclature I mentioned about is the water leaving radiance over the downwelling irradiance. And that's what we usually call remote sensing reflectance, which is actually what it's one of the products of what we can obtain from remote sensing. And this can be, as I mentioned here, related to backscattering absorption of the total backscattering and total absorption that would happen in the water. So this is, so basically, as I mentioned before, the color actually is the spectral variation of the reflectance at the water surface and the spectral variation of this reflectance. So just as an example, simple example to see, to understand this concept, we can talk about water. So the main part of the water body is water in itself. So basically this is the absorption, how it looks, the absorption of water in the visible region. So we see very low absorption in the blue and increasing towards the red part of the spectrum. In turn, the backscattering of the water have this shape. So its maximum is higher at the blue wavelengths, around 400, and decreases towards the red and more in the infrared. So if we have this approximation of the reflectance being the backscattering over absorption, we can see the reflectance of the blue of the water, just water, have a peak in the blue because the relation between absorption and backscattering at a given wavelength. So this is just an example of this relation and a simple case, which is the water. So, okay, we know how the water reflectance looks like, just pure water, but we see actually what we want to see is the change in its color. So because there are other constituents that change the color, that's why we call them optically active substances in the water, because they have this capability of changing the color of the water in the visible part. So for simplicity, in general, the constituents of the water body can be divided in four. One we call water, we have seen it. Here's like a sketch, schematic view. The other part we call phytoplankton, which actually is pigmented microscopic algae, so mainly having chlorophyll A, which is a pigment present in all the plants in land and water. Also, what we call suspended material particles or non-algal particles, because these are particles suspended in the water. Phytoplankton too, they're particles. But in this case, we separate if they have pigments or not pigments. So what we call non-algal particle is actually particles that don't have this, are not pigmented, basically. And the fourth one is what we call color dissolved organic matter, or CDOM. In the literature, you will find other names, yellow substance, gelbstoff, gilvin, but basically is a difference to the other part is actually dissolved. It's not particle because of the size. Arbitrarily, it's defined as dissolved and not particle suspended sediments. So here's some definition that it's very useful to have in mind, and when we talk, is what we call about the inherent optical properties against the apparent optical properties. So basically what I've been talking about, absorption, scattering, they're both inherent optical properties. And the characteristic is that they only depend on the type and concentration of the substance. Here, well, I added the attenuation, which actually is the sum of the absorption plus the scattering. That's what, so how the light is attenuated is the sum of how much is absorbed plus how much is scattered. So this is, in turn, the apparent optical properties actually depends not only on the type and concentration of the substance, but only on the illumination geometry. So something external at the substances that are present in the water. Example of this is radiance, reflectance, what I've been talking about before, because it depends, as you can see, it depends on the angles of viewing and also the wavelengths. Reflectance, which I shall mention later, before, sorry, and for example, the diffuse attenuation for the Welleny radiance, KD, is how the light is attenuated with the So, as I mentioned before, reflectance can be expressed as a function of these IOPs. Now we know it's absorption and backscattering. So basically, it can be expressed as a relations between the total backscatter of the water over the absorption plus the total backscattering. So the interesting thing that these IOPs have these properties of what we call additivity, that it means that the total inherent optical properties can be calculated or estimated as the sum of the IOPs of each of the components present in the waters. What it means, so total absorption can be expressed as the absorption of the water plus the absorption of the phytoplankton of the four groups I mentioned before, the non-algal particles, and absorption of sedum. And the same for the backscattering, the other IOP I've been talking about. It can be the total backscattering can be expressed as the backscattering of the water, the phytoplankton, and the non-algal particle. By definition, and the size of what can be defined as sedum, it's assumed that it doesn't scatter the sedum. So it will not appear here in the total backscattering. So finally, reflectance in this way can be expressed as a sum of all the components, backscattering and absorption in this way. So actually I will go, so what we want or what we see, what will tell us the color, it will the variation of this reflectance, which will depend on all of these components. So I will go through the four components, water, phytoplankton, non-algal particles, and sedum, and looking at what are the optical, the absorption and backscattering, how they look like, the spectra that will give us the result of the reflectance. So we start with the water optical properties. I more or less show you before, but I'll go a bit more in detail. So basically, the absorption of the water body is generally considered invariant. So it's tabulated. You can find it in different references. So basically, it dominates in the red and infrared, and minimum in the blue. Here, the spectra is quite wide because it comes from 100 to 10,000, and the visible is only this part. So if we look into more detail in the visible part, has the shape I showed you before, very low or minimum in the blue, increasing toward the red. And it's a part of this interesting characteristic of the absorption. It acts like a filter with depth. So following this spectra, we can see the red and infrared light is really highly absorbed. So it's mainly absorbed in the first millimeters of the water. And then the absorption decreases. So, and being minimum in the blue, it means it will reach deeper in the water column. And then starts to increase in the violet and UV, which is not shown here, but it wasn't here in the previous. It started increasing in the UV. Backscattering the water itself, only water. So it's really low, especially in the green and red part of the spectra. It varies with lambda to -4, around -4. And the other particular thing is that it's symmetric respect to 90 degrees. So this means lights coming this way, interacts with with water molecule, let's say. So the light is scattered in all this direction in a symmetrical way. So basically can be expressed that backscattering of the water is half the total scattering of the water, which for some things simplifies a lot of the equations and so on. Next, the phytoplankton. So the absorption, I will talk about the absorption and backscattering of phytoplankton. Basically, what we call phytoplankton absorption actually is the sum of the absorption of all the pigments that are in the cells, in the different species, whatever you have in the water body at this time. So you can see in this plot that actually you have this is the absorption and wavelength index. And you see that for different pigments, here you have chlorophyll A, which I put it in green, chlorophyll C, chlorophyll B. So you can see actually there are different pigments that have different spectral characteristics. And the sum of all the pigments, it gives this dark, bold black line. So basically what we see is that there's a peak, main peak in the blue, and another peak in the red of the total. But actually, if you see the chlorophyll A is the main absorber in the blue. So this peak generally coincides with the chlorophyll A peak in the blue. and a second, the peak in the red. But just to show you that this is highly variable and depends on the species. So species can have different pigment compositions. And here you can see different colors, different species groups, let's say, not species, sorry. But actually the common thing to all of them, you can see, so these are the absorption of the group, the total absorption. So it's not just one pigment, but it's for each of the group, it's the sum of the absorption of all the pigments each of this group has. So basically, but what I want to show is the high variability between them. But actually what they have in common is that all have the peak in the blue and a second peak in the red. And that's what is generally useful, or it's used for determination of chlorophyll concentration using remote sensing. Okay, in general, is the main absorbing constituent in the more open ocean or what we call oceanic waters. While in the more coastal areas, usually you have other like sediments that also contribute to the total absorption. Okay, so the backscattering properties of phytoplankton also is the main substance that scatters in this open ocean, as I mentioned. And usually it's modeled as an exponential function respect to wavelength. So this exponential form, backscattering in wavelengths. And also it has been modeled as a function of chlorophyll concentration. So how much is scattered, it will depend on the amount of chlorophyll A pigment. That's the idea of how to estimate or be able to model the backscattering coefficient of phytoplankton. The third group is the suspended material or non-algal particles. So these are particles that don't have pigment, or what we saw. And in general, it's mainly inorganic, so sediments, which reflects more or less the geologic or the earth that is adjacent to the basin you're looking at. But also it contains its part, it has an organic part, sorry. So basically, it's usually generally assumed to have biological origin, which means part of animal, the phytoplankton fragments, mainly organic bodies or dead organisms, fecal material. All this goes to what we call non-algal particle or suspended material particles, basically. So the absorption characteristic of the non-algal particle is that the absorption is high in the blue and decreases exponentially towards the longer wavelengths. And usually it's modeled in this way. So this is the exponential, where the S here is the slope. And in different references or studies, this slope have shown to have some quite a variability ranging 0.06 to 0.13. So this is how it looks. And the difference, you can see this absorption, it doesn't have the peak, which is characteristic of the pigmented particles. In turn, backscattering also can be fitted to an exponential function, and the exponent also has shown to be variable. And actually, this depends on the size of the particles. So it has shown this exponent vary between lambda to -3 to lambda to -0.8. So it's quite variable, that can be fitted using this model. And we go for the fourth component, which we mentioned it was the color dissolved organic matter, or CDOM. And what is it, the CDOM? Okay, CDOM is a complex of mix of organic compounds of humic and fulvic acids that result from terrestrial and aquatic vegetation. So if you see when you water plant, I don't know if you ever seen you water plant and you see the water coming after passing through the earth, and you see this yellowish color of the water. That's basically also what we call yellow substance, because it's mainly dissolved substances that have an organic origin. And also it can be local or external, depending if in the sea, we are talking now about water bodies, if this come from degradation of the phytoplankton cells or other organic particles, or can be external if it comes from river, for example. As I mentioned, the plants we water, when coming from the river, it will usually have sedum that come from all the basin that has been draining all the water. So in some cases, it has been found to be correlated to chlorophyll A, and in other cases to salinity. For example, if it comes mainly from rivers. So it depends on the place of the basin. Another interesting thing is that sedum can suffer for the degradation. And as I mentioned before, it's a dissolved substance, so it assumes that it doesn't scatter light, just absorbed, and can be the main absorber in coastal areas, which are mainly influenced by rivers, because they come from the river in itself. So the optical properties of sedum, the absorption also is mainly in the blue and follows exponentially. The exponential function increasing toward the UV, and also with this exponential that has been found to be quite variable too. I will not talk about scattering because I mentioned it doesn't scatter. So basically, how we go from this substance to IOP to reflectance, what the relation. I mentioned that we can separate the substances in 4 groups, like chlorophyll, sedum, and non-algal particles. They have their own absorption and backscattering signature shapes. And following, like what we call a direct model, we can get to what would be the reflectance of this water, considering all the substances that are present in the water. Well, actually, what remote sensing is, actually is the reverse model that we need to find. So we will have the remote sensing reflectance spectra, so which shows the color, and we want to retrieve what actually is in the water. So what are the substance and in what quantity, the concentration of each of them. So basically this is the job or what algorithms do. So basically the algorithm or the inverse model is what we use to estimate the physical values. So this is what I call the direct, what I've been going through with you. And the inverse models can be either empirical, so from reflectance, we can find statistical relations with the substance present in the water, with a given band or boundary ratio, just an equation. Or we can go through the IOPs, or what we call the semi-analytic inversions, which actually there's some relationship, and part of them are empirical between reflectance and IOPs. And finally, is another type of product which use the multispectral fitting. So basically use all the bands, all the spectra to fit all the components and use different statistical methods like neural network and so on. So I will focus on algorithms of phytoplankton, of chlorophyll concentration, which is you will see it will be mainly used for all the applications and the coming modules. So we'll focus on the standard algorithm, just a description of what it means. And so we'll went back again towards phytoplankton. So these are microscopic single cell photosynthetics. So they have this pigment that allows them to do photosynthesis, and they live in the water suspended. So like plants, so they take carbon dioxide from the atmosphere, transforming into organic compounds, and in using light energy and pigments. finally releasing oxygen. So they form the base of this of most of the food chain. So that's what they call primary producers. So why they're important and when we study them, because mainly they affect the distribution of and abundance of fishes, and they influence the cycle of elements as I mentioned, and you can see oxygen and carbon. In this way they influence the climate system. You will see in future modules of this series of webinars, and they contribute approximately to half of the Earth's primary production. So it's very important to study. And how we study, how we have known the amount, how we can estimate. So one way I put here just pictures of different, so we can see the high diversity of cells, shapes, size. So it's really, we call phytoplankton, but actually it's really variable group, let's say. So one way of estimating them is counting cells. So cells per volume, take a volume of water and count the number of cells. We can do it with microscopy. So looking at the microscope, flow cytometry, which just is a flow of water, which automatically counts particles in general. We can estimate the amount as the carbon content, and also using what we call the chlorophyll concentration. So it's not the only pigment present, but since it's present in all of the groups, it can be a proxy of the amount of phytoplankton present in the water. And for this, we can use fluorometry in the lab. So using the fluorescence capability of the chlorophyll, or we can also estimate it from remote sensing, as I've been mentioning, using this ocean color, this sensor in the visible part of the region. So for doing this remote sensing, you have different type of algorithms, but in general, groups can be. One is the most used, especially in open and ocean waters, the blue-green ratio, which I will talk about. Going to more coastal or turbid waters, they don't work. You probably will see it in the later modules when they talk about coastal waters. But usually there you use other bands, the red and infrared part of the spectrum. And then you have this other type of algorithm that use all the spectrum to retrieve the chlorophyll concentration. So I will just mention and talk about the blue-green ratio, which is the most used, and mainly the standard product of all sensor is the chlorophyll concentration for open waters. So basically, this algorithm uses the optical characteristic of the chlorophyll that has a minimum in the green and a maximum in the blue. So, which I showed you before. So if we can see how the water reflectance changes when the chlorophyll concentration increases, you will see, given the absorption in the blue, the high absorption in the blue, when chlorophyll concentrations increases, the reflectance will decrease in the blue, while in the green, given it's almost constant, basically it will stay the same or slightly increase. So taking this ratio between the blue and the green, it will give difference in this slope, and it's basically related to the pigment concentration of this ratio of these two bands. And that's what the algorithm is about, has been developed fitting large amount of data collected all around the world, and by calculating the ratio and fitting this equation to the measured data. So this is reflectance measured in situ, and chlorophyll concentration also measured in situ. And these are what has the relation that has been measured, and the line is the best fit of all this data. So basically and this is done for different sensors you have maybe already heard about. So we'll go a bit more on this sensors. So I talk about what's ocean color, what's the color of the water, and what does it depends on the substance present in the water. But now I go back to what we started, which is remote sensing. So what we call ocean color remote sensor. Basically, these are sensors that have bands in the visible part, because we need to see this changing in color, so in the visible, so we need bands in the visible part. Here are typical Modis Aqua, typical what we call ocean color sensor, beers, Sentinel-3, and on the right you can see the spatial resolution of these sensors, and you can see all of them have bands in the blue, in the green, in the red, and also in the near infrared. But apart from the traditional ocean color remote sensors, we also have what actually we've been using also for ocean color is what has been actually defined for land applications. But since they also have these bands in the visible, and okay, sensors have been changing, and so now they're thought already to be useful in the water system, but they also have these bands in the blue, green, and red. And as you can see, have better spatial resolution, 10 meters, 30, 60 meters for another type of use, more coastal, useful in coastal areas. But they also have this part, the near infrared, bands in the near infrared, sorry. And more lately, it's what Prisma, for example, is it's a hyperspectral. So you really have more really large amount of bands, and that's so it actually also covers the visible part and also the near infrared. And as I mentioned before, okay, so the visible bands are used, as I mentioned, to detect the change in color, basically, but we also have these near and sweep bands of all the sensors. And that's not random. I mean, they have their use. So basically, when we have a sensor, it's usually these are around 700 kilometers away from the Earth. Basically, it's detecting the information that originally the energy comes from the sun, interacts with the atmosphere, then with the water and surface, then back to the atmosphere, interact again with the atmosphere, and finally get to the sensor. So basically, we have something in between, which is the atmosphere, and which introduces kind of a noise, let's say, so something signal we don't want. And these bands are mainly used for this correction that we call atmospheric correction, so get rid of the atmosphere. So this is just to summarize what I said. So what arrives at the top of atmosphere, so the radiance that arrives to the sensor comes from what the light interacting with the atmosphere is scattered and absorbed. Part is scattered and absorbed. Then part interact just with the surface, like the sun glint or the specular reflection. Part of the energy will enter the water body and will be absorbed and scattered by all the constituents, as I mentioned, and finally go back to the sensor. And also, if it's shallow, if you can see the bottom, if can reach the bottom, sorry, the energy, the final signal arriving to the sensor will have information from the bottom. So basically, the reflectance of the water is what we are interested in. So from the total of the radiance that arrives to the sensor, the information we need, or we can get information from, is what's coming from the water, not the rest. So basically, the idea is the rest will be what's coming from the atmosphere and surface. So the objective of the atmospheric correction is to identify and remove the component coming from the atmosphere and the surface, to retrieve the water coming from the signal. The thing is that actually it's very low, so almost depending on the wavelength, but the contribution of the atmosphere can be really high. So the water signal is quite low. So this part of the processing is important. As you can see, if this is the radiance that arrives to the sensor, you can see there's almost no shape. So we said that we can get information what's in the water, looking at how this reflectance or radiance varies with the spectra, but we can see that actually the total radiance that arrives to the sensor has not really shape. While if we subtract what comes from the atmosphere, you can see that the clear water reflectance will look like this, so have a peak in the blue. And another case, if the water is green, you can see both the reflectance in the green, which tell us there's more green water. So basically, this correction of the atmosphere is very important to really get this information from the signal. Otherwise, we cannot see it from the top of the atmosphere. And from all these components, so the total, you can remove the atmosphere and the surface. You have to remove them. What's coming from the atmosphere can also be divided from what comes from the molecular part of the scattering and the aerosols. But the important part is actually the Rayleigh molecular scattering can be calculated, it's tabulated, depends on wavelength and geometry. So that's kind of the easy part. But actually what I want to focus here is that the most variable thing is the scattering of the reflectance coming from the aerosols, which can vary in space, time, and size and type of aerosol. So basically the size of the particle. So, and what I want to mention is the typical or standard atmospheric correction, which is called which uses the black pixel correction assumption, actually. So, in a few words, this atmospheric correction, what it uses is the assumption that the reflectance in the near infrared is 0. As I mentioned before, one of the characteristics of the water absorption is that it's really high in the near infrared. So basically, it's really absorbed. As soon as the light arrives to the water, it's mainly absorbed. So actually, if the sensor is receiving a signal in the near infrared, that means it's not coming from the water, but from the atmosphere. So back to the previous equation, we can estimate the component of the aerosol from knowing what's coming from the water. So we have the total arriving to the surface. This can be to the sensor, This is from the surface that can be calculated. The Rayleigh that I also mentioned, it was can be also calculated. And we assume that it's really clear water, let's say. So the reflectance from the water is 0 because of this characteristic of the water that it's highly absorbing in the near infrared. So knowing this, we can estimate the absorption of the aerosols or the reflectance of the aerosols in the near infrared part, and then extrapolate it to the visible part and correct for the aerosols, knowing the shape and type of aerosols using these bands in the near infrared. So that's the basics of what we need for atmospheric correction. So basically, the ocean color reflectance, we have the radiance or reflectance that arrives to the sensor. We need to apply the atmospheric correction to get the reflectance or water radiance that I mentioned then. And then we apply biooptical algorithms. So the idea is to have this equation that gives us the concentration of this optical active substance that we're interested in estimating. So in general, this top of atmosphere radiance or reflectance is called level 1 in the images you will get, while after the atmospheric correction, this water reflectance or radiance and the concentration or different products usually are within what we call level 2. So it needs a processing, which is the atmospheric correction at least. So this can be found in data repositories, which will be a topic of the future model by Ben. So this you will get where you can get this data basically. And also, to get from one level to the other one, you need to process them. And also there will be models talking about processors like Snap, CDAS, or you can do things in Python. Basically, these are processors that have been developed by the space agencies, some specifically for their own satellites, but some of them are more wide use and can be used for different sensors. And usually each space agency define the standard product for each of these level 2 data. I know Lorraine also have been talking about the level of processing, and she mentioned that it depends on the, how they call it, level 1A, level 1B, level 2. But basically level 1, it means there's no processing, just the data top of atmosphere, that's what I mean, sorry. And level 2 is already processed with kind of atmospheric correction and at the right product. So to sum up a bit about this webinar, basically what's ocean color remote sensing is the variation of the spectral form and magnitude of the water reflectance in the visible part. That's what relates to color. And basically it's used to derive quantitative information of the type and concentration of this substance that are capable of changing the water color. You cannot derive any substance you would like, but if they change the color, you might be able to estimate them. The spectral form is basically determined by the IOPs, mainly absorption and scattering of the different substances. And it's an important tool for monitoring both from the phytoplankton in the global ocean to monitor water quality, the ecosystem status for long time scales related, for example, to climate. All this will be subject also to future webinars. And I mentioned also the atmospheric correction, which actually is quite essential to get really accurate estimation, because if you have really high errors in this part of the processing, you will not be able to trust or have a good estimation of the derived products. And that's it. If you have any question, I hope it was clear enough.
Thank you very much, Anna. I'm sure there'll be plenty of thumbs up and applause, but yeah, that was a very nice overview. So there's already been a number of questions come in. I will give it a few minutes because we always seem to have a lot come in at the end of the lecture as well. So please do feel free to put all the questions you would like into the chat. I will ask, please refrain from putting too many or putting links or anything like that in. But yeah, all questions are welcome. And while those are coming in, I'll just answer a couple of technical questions that came up about the webinar series first. And then we'll move on to the kind of panel discussion around ocean color. I was also wondering if the panelists wanted to introduce themselves that haven't already been introduced, maybe Shuba, Osvaldo, Gemma, because it could be that there's answers coming from all across the panel. So maybe we'll start with a panel introduction. So obviously you've just all heard from Anna Dugworthy. I'm Thomas Jackson, so I work at Plymouth Marine Laboratory, and I'm a senior remote sensing and ocean color scientist. Gemma, do you want to go next?
Yes, definitely. I'm Dr. Gemma Cook. I'm also a senior scientist in Earth observation, but also phytoplankton physiology, primary production at Plymouth Marine Laboratory.
Osvaldo?
Hi, Ana. Hola. I'm Osvaldo Ulloa. I'm from the University of Concepcion in Chile, but I'm doing a sabbatical here at PML.
Shuba?
Hi, I'm Shuba Satyendranath, an oceanographer, and I have been working for many years on optical properties of water and its applications for ocean color remote sensing. and for computing primary production in the oceans. I have been a long-term student of Trevor Platt, and I work at the Plymouth Marine Lab.
I don't know if Stefano wants to introduce himself from ESA as well.
Thomas, thanks a lot. I'm Stefano Ferretti from the European Space Agency. I work in the Science Applications and Climate Department, and I'm really glad we have been able to organize this capacity building activity together with Plymouth Marine Laboratory and in particular the Trevor Platt Science Foundation. Thanks.
Okay, so on to the technical questions about the recording and access to the materials. So there were a number of questions around, will the notes, the slides, or the presentations be available after the course? The intent is, what we're doing is we're recording the lectures and these question sessions as they happen. And then we upload basically a moderated version of the video to YouTube. That takes some time, which is why the first module is not yet available. It's in processing with YouTube at the minute. The channel for those will be the Trevor Platt Science Foundation channel on YouTube. So I think you can find that with YouTube tag @Trevor Foundation. So please go there, and if you subscribe, I guess you will get the notifications when the videos are uploaded. But that will happen in an ongoing manner through the series. So from that, you'll be able to see all the slides again and go back in case you missed something or want to hear something explained again. Okay, so then on to the, yes?
Tom, could I make a request to all the participants before we go into the question? Yeah.
Go ahead.
Okay. I've been watching people coming into the session, and I was a bit disappointed to see that most of the participants came very late into the lecture. So if you are serious about this course, and if you would like to make the most of it, it is very important that you come in at the beginning. Otherwise, you won't get everything that people like Anna are trying to tell you. So we have tried to make the demand on your time not too severe, so that you only have to devote about 2 hours every week to this lecture course. But please make sure that you do set that time aside, and please try to come in on time. If you do that, you may find that you will need the recording a little bit less, because if you listen to the lectures in the first time, maybe you won't need to go back to the recordings. And if you come in late, you lose the flow, you miss the fundamentals, and you will struggle to follow. This will be the same for all the lectures to come. So I really would like to request that all the participants take the time to come to the lecture on time. I am an old-fashioned, and I am also a very old person, so you will forgive me if I say that it is also a mark of respect for your teachers. So please consider that seriously. Thank you very much.
Thank you, Shiva. Okay, so I think I'll try to have these questions follow the rough order of the lecture, beginning with some of the technical points around reflectance itself, and then we'll move on to derived products. But we may jump back and forth a little. So I think there was a number of questions around the reflectance data that's used. One of them was asking about Sentinel-3 data and if the reflectance data that is available is dependent on geometry or if it's normalized. So that's, I think, Anna, as far as I'm aware, the sentiment, well, I mean, this depends on how you've atmospherically corrected the data. Some atmospheric correction schemes will produce a normalized reflectance, and some don't apply that. The default one coming from ESA through Sentinel, the Sentinel-3 data providers, is normalized reflectance, as far as I'm aware. I don't know if you want to correct me on that, but that's my understanding.
It has not for the moment. They are thinking about doing this correction of BRDF. So actually, it's not corrected.
Okay, so it's not fully normalized.
Not
So
for the moment.
Okay.
They're thinking about doing it, but not yet. For the moment, it's not.
Okay. So the default level 2 coming from Sentinel-3 has an angular distribution. But if you, for example, as we will show later in the course, if you process using something like polymer, that will produce a normalized reflectance. So there you take the level 1 and you produce the normalized reflectance. But the level 2 that is provided has all of the atmospheric correction applied. So there's no, effectively you're viewing it without an atmosphere, right? But it's just got that angular distribution. Okay. Okay, so there's another question which says, what would be the maximum depth of using remote sensing to understand what substance makes ocean color appear as it does, or the color of the ocean? I guess this is related back to optical depth, which Lauren covered briefly in her talk, but maybe you want to expand a little on that.
Yeah, well, actually, the optical depth, so at what depth will actually reach and have the, actually what the satellite sees is an integration of a certain depth. It will basically will be determined by the amount and type of sediments, particles or substances that are in the water. So if really clear waters like the gyre, the snow, it can reach really high meters of depth, let's see. So it will be, and when you have more substances, particles, absorption, it will vary the depth of how deep the sensor will actually have information from.
Yeah, I think when we discussed optical depth last week, we said it could vary from centimeters in very turbid waters to tens of meters in very, very clear waters.
It will also depend on the wavelength, right?
Yes.
Yeah.
So even in clear waters, the red wavelengths won't penetrate very far.
Yeah.
There was a question. I'm not sure this is, I mean, it is remote sensing. So there was a question about atmospheric correction and drones or UAVs, and is that required? I mean, there's certainly some corrections which are common to all remote sensing. So again, viewing angles and things like that. So if you're trying to get to the IOPs, what you're actually measuring is the AOPs. So there's definitely some corrections that have to be made, whether it's from a satellite or a drone. But I don't know if you want to add anything on that one or not.
Yeah, as far as I know, I mean, the drones are not that high, so you don't have to do like the atmospheric correction complete as because it's not enough measurements to have that kind of influence, but it needs some corrections for the angle and all these characteristics. It's not the same. I don't know how it's done actually with drones, but it needs some corrections, yes.
While we're on the topic of IOPs and reflectance, there was a question about which satellite products are most recommended to calculate IOPs or reflectance, especially chlorophyll and CDOM. And again, you can expand more, but I think that would the satellite you use depends on the application. This is something Lauren was quite emphatic about, because it depends on whether you need a certain spectral resolution and what spatial scales you want to observe at and these sorts of things. But there's a range of ocean color satellites that we can recommend.
Yeah, I got lost with the question. It means which sensor can be used for different.
I think so. That I would summarize the question as which satellite products are most recommended for estimating reflectance or IOPs. So I guess for that we would recommend
it depends
Ulti,
on
MODIS, MERIS, you know, depending on going back through time, you know, all the way back to CWIFs. You know, we have decent sensors with a good accuracy, but yeah, currently flying for large scale ocean applications, Sentinel-3 has good coverage, spatial coverage. Sentinel-2, I mean, I think if you go back to one of your slides where you had all of the bands, it's quite important to show the difference in the width of the wave bands that you have, because some of them have quite small. Here, if you look at the difference between MSI and ULCI in the blue, we have very different bandwidths. So here you might recommend ULCI if you're looking at trying to derive a spectral reflectance.
Indeed, yes. But as you said, also the application, maybe you need to use in really coastal and you really need higher spatial resolution, and ULCI is not good enough. So you go to what you have, which is Sentinel-2. But yes, that's it will basically also influence, be determined of the use and application you need.
There's been a question about whether ocean color and chlorophyll approaches for phytoplankton abundance might be affected by photoacclimation. Would you like to discuss, or anyone on the panel to contribute to that?
Tom, could you repeat the question, please?
So the question is how ocean color and chlorophyll estimates of phytoplankton abundance might be affected by the issue of photoacclimation. So for those that don't know, photoacclimation is the process where the high light phytoplankton basically change their pigment and optical characteristics. So things like fluorescence will definitely change under high light.
Want me to have a go at it, then everybody can pitch in and help?
By all means, sure.
Okay, so the question is whether the photoacclimation has an effect on the algorithms. I don't know if I am reading too much into the question, but taking it at face value, and if we are talking about the algorithms for estimating chlorophyll A, then we can think of the possible situation that if phytoplankton are adapting to low light, provided low light appears at the surface, then photoacclimation would lead to increase in the intracellular concentration of chlorophyll A. There is an optical principle that relates the efficiency of absorption by any substance when they are contained inside a finite particle, that the efficiency will change depending on the size of the particle as well as on the intracellular concentration of the particles. If the concentration is increasing, the effect would be to reduce the absorption by the chlorophyll concentration compared with the same amount of pigment if they are in more dilute concentration inside the cells. So in principle, the effect could be that the absorption in the blue part decreases due to increased acclimation to low light, and then you might interpret that as lower chlorophyll concentration. But bear in mind that for this effect to happen, it has to happen at the surface and at noon time, more or less, when the satellite is passing. So usually this effect would add to some uncertainty in the estimated chlorophyll concentration. But similar uncertainties can also appear when phytoplankton cells are of different sizes. Small cells have higher absorption efficiency. Large cells have lower absorption efficiency. Now, if by talking about photoacclimation, you mean the relationship between chlorophyll and carbon, then that's another problem altogether, which Anna hasn't addressed in her lecture. She's been talking about algorithms for chlorophyll estimation. So it would be irrelevant to this part of the lecture, but it could have relevance for certain types of models of primary production that depend on knowing the carbon content of phytoplankton rather than the chlorophyll concentration. In that case, you need to make extra steps when you convert the chlorophyll estimated to carbon concentration, or you have to use another set of algorithms altogether to estimate carbon from space. And that too, not any carbon, you need to estimate carbon contained inside phytoplankton. Hope that answers the question.
For more details on carbon estimates, I guess we could direct students to the BICEP project.
Sure, yeah. And also, you will find that if you go, Gemma, could you type the website or something for BICEP project?
Yeah, of course. I was also going to mention, I'm pretty sure we'll discuss more of these things in following lectures during the training course.
That's right. There is a module that is devoted to climate and carbon and primary production and so on. So these matters will come up in more detail there. So if Gemma is posting the BICEP website, you will find a number of products and publications that are relevant to the question. But we are not the only ones working on the problem. If you search on the web, you will find even more publications that talk about this particular problem of photoacclimation.
So while we're on the topic of things that make it difficult, there was a number of questions around different phytoplankton groups, which Anna showed some beautiful pictures of. And if you could, it would be nice to hear your thoughts on which different groups we can identify from ocean color and how we take those differences in community composition kind of into account.
Okay, so for the general algorithm of the chlorophyll I mentioned, actually it's just this band ratio, and actually it's not species specific. So to get more information about composition, you need to, so probably not any group or species could be able to be distinguished. Mainly what has been published and working is general groups that have specific pigments that can be used to differentiate specific group from others. Classic is coccolithophores that has a specific, these are platted cells. So they have cells that have plates of carbonate calcium, so they reflect a lot of light. So they have this spectrum, which is really characteristic. You can see in the remote sensing images like turquoise, so have specific spectra that is quite different from other phytoplankton, but also similar maybe to sediments, which is another problem. Work has been done differentiating cyanobacteria. For example, cyanobacteria have a specific pigment called phycocyanin that have an absorption in a band like 620. So it's specific, this pigment, for this group. So there has been algorithms that try to detect this absorption in the 620 band. But what will be determinative if we can differentiate or not also depends on the sensor. So if the sensor has that band, a band located in where the spectral feature of a certain species has this feature, if it doesn't have it, if the band is not there, you will not be able to see this change in reflectance due to these specific pigments. For example, so that's one main. Also, there's been working about differentiating diatoms from other, and sometimes it helps knowing what's the typical or the dominating phytoplankton community in your region. Sometimes, so in my region, you can find diatoms and cyanobacteria or chlorophytes. So there's some basic knowledge on your side that might help you try to differentiate these specific groups, with external knowledge and measurements of absorption that can be used to tune the algorithm locally to try and differentiate. But mainly also it will depend on the sensor use, which have the right bands with the right width of the band that will help you to differentiate them.
Okay. There was a question around how we go about validating atmospheric correction. So, I mean, I can say we go and take measurements all over the world. There's a number of dedicated stations called Aeronet, which measure surface reflectances. I mean, Anna, if you've got experience of doing this directly, so maybe if you want to expand, but
yes.
Yeah, we do it often with in situ. Yeah.
Yes, actually, for the validation of atmospheric correction, what we need, so we will have the reflectance or what's measured by the satellite. We have an algorithm that will give you a surface reflectance, and we go to the field and try to measure the same as this satellite is measuring, basically, and compare what's the reflectance we measure in the water to what after the atmospheric correction you get from the satellite. And as you said, from the in situ measurement, you can go with a ship, for example, with a radiometer, either above water or in water can be put the radiometers. There are different techniques useful for different type of waters also. But also we have these automatic sites that actually they're fully automatic. So they made, you don't have to be there actually making all the measurements. You mentioned Aeronet OC. There's also now more HyperNet, Hyper Water. So it's similar. It's the same concept that the Aeronet OC, but with using a hyperspectral sensor, which also gives more information because it's completely automated. It's measuring all the required parts to derive the water reflectance. And finally, compared to the satellite, which should be measured similar or close to the passage of the satellite you want to validate.
Okay, there's some questions. So in your diagram of all the various components of light that actually end up being detected by the sensor, one of them was the seabed. And in a lot of cases for ocean color applications, we might mask, for the open ocean, we might mask waters where we see the seabed because it's considered a contaminated signal. But some people have asked about, are you able to map seagrasses or different habitats or anything like that? I'll let you speak first, but yeah, there is some ongoing work for that for other projects as well.
Yeah, no, exactly. What I know or have been reading is that it needs a lot of modeling, but there has been many efforts to try to get information from the bottom here, this one. So yes, you did quite some modeling because you have what's in the water apart from what's in the bottom. So it's not an easy, but it has been really an issue for study.
I know one of the there's a project looking at seagrasses, and part of the approach that they use there, so they've trained a machine learning algorithm, but also part of the training and identification of seagrass areas was done because obviously things in the water move around and change a lot from day to day, whereas the seagrass is fixed to the seabed. So what they've done is they've looked over a longer time span, and effectively that allows you to remove some of the noise of the water in the atmosphere because the benthic component that you're trying to retrieve is relatively static. Obviously, the seagrass grows and blooms and develops, but that is one approach that you can use when you're trying to map, let's say, static seabed or land features compared to dynamic water or atmospheric features.
Tom, there was a similar or related question about detecting corals and coral bleaching in particular.
Yes. So, is it possible to study coral health in shallow areas for the purpose of coral bleaching monitoring? With some of the high resolution satellites, you might be able to detect a bleaching event, but the hard part, well, I mean, by all means, the rest of the panel can step in. The hard part is that often when things turn very bright white, when we're dealing with ocean color signals, they get masked. There's a lot of flags, and so you will need to start at quite a low level, so maybe at the level 1 data, and provide and start processing from there, because you'll need to make sure that you aren't applying all of the default masks to the data, because otherwise you might remove your signal. So yeah, there is potentially a signal there you could see, but you would need to start probably with the level 1 data and look at how you're processing that.
Yeah, and maybe I can jump on that, maybe not specifically to coral, but just as a remark in general, because we use, okay, we have the level 2, it's already chlorophyll, it's enough. But maybe since I mentioned these are specific like clear water applications, so it's valid for really clear water. So it doesn't mean it will work everywhere. And probably if the region is optically complex, you will see like highly turbid or really like, as you mentioned, when you have this bright coral bleach, then you get these things that you get masked and you say, but I know there's something there. And so for more specific, more complex water that probably in the module of coastal water you will need a bit with, there is a need, you will need to go back and up in the level to have alternative processing. So there's a lot of people working on complex water, but you have to know that probably the standard output will not work.
Yeah,
But
so
it's part of the complexity.
Factors like the dark pixel correction, where you were saying you assume zero reflectance in the near infrared. In
Exactly.
very, very turbid waters in the coast or lakes, that might not be the case. We had a question about if these methods can be used for other aquatic ecosystems or just for the ocean. So in the broad sense, yes, you can use color remote sensing to work with complex coastal waters, as you will see from Son's module coming up. You can also use it to look at lakes and inland water bodies and rivers. There are dedicated projects looking at that. ESA has a climate change initiative project looking at lakes, which does not only ocean color, but also looks at temperature and a number of other variables. So yes, you can definitely use these principles to look at other aquatic systems beyond oceans, but the complexity increases and you have to take a lot of care. So, you know, taking what was said in this lecture, you have to really check all of your assumptions when you move into the more complex waters. But yes, it's definitely, there is ongoing research. Okay, we have a couple more questions around water constituents. So there were some questions around CDOM and telling the difference between well, humics and fulvics was one question about if they have different signatures, or can you tell the different proportions, or is it all bundled together? And there was another question around coloured detrital matter versus coloured dissolved organic matter, and whether or not is it possible to convert between them, or are there empirical relationships? So I think if you could just go back to your section on CDOM and expand a little there, that would be very good.
Yes, well, I guess so. The question was if we can differentiate the different humic or fulvic acids. That was the question. Okay, so what I know that not exactly, but you can find. I know I read papers that they differentiate the origin of the CDOM looking at the spectral slope. So the S depends on. So that's kind of they found that it was related to the origin of the CDOM part of the basically related to the slope. So you can see it can change, it varied a lot, and in some regions, or making like a big comparison going from really oceanic to really coastal waters. I know there was a work in crossing the Atlantic, so they found this changing in slope related to the origin of the CDOM. It's not okay, they say this is humic, this is fulvic, not so precise, but at least you can differentiate the origin of the dissolved matter. That was one, and the other one, maybe I lost.
The other question on CDOM was asking about differentiating between coloured detrital matter and coloured dissolved organic matter. So I guess that's the dissolved versus the particulate components.
Yeah, well, that's the most difficult. Sometimes some algorithms just as a result, given that they're really similar shapes, so slope, you can see the absorption decreases in a similar way. You just name different the slope. So actually, from remote sensing, it's really difficult, not possible actually, I think, to separate both. So actually, what as a product you have the detritus absorption, which is a mixture of the result of the absorption of CDOM and non-algal particle absorption.
Yeah. So I know with some models, they output what called ADG. So the absorption of detritus and yellow stuff together. Yeah, because it's hard to distinguish. Yeah. On the topic of distinguishing between signals, there was a question around differentiating particles of the same color. For example, an algal bloom is green, and so is water hyacinth. So how do you distinguish between them?
Water hyacinth is the floating, right?
Yes. Yeah.
Okay. Well, that makes it, that's kind of easy, let's say, because it's a water plant already floating, so it's above the surface. And actually, looking at the reflectance, I don't have it here, but actually it looks like a terrestrial vegetation. You have a big signal in the near infrared caused by the vegetation, which is above the water. As soon as it's vegetation inside the water absorbs, so you have really low reflectance in the near infrared compared to when the vegetation is floating or above the surface. You get signal in the near infrared, and you can easily, let's say, differentiate if it's floating or above the surface while it's submerged or within the water body.
Yeah, so in the green section they may look similar, but in the near infrared they look very different. Yeah, so it's again about choosing the right bands to differentiate. There is a question around if we can spot plastic litter from the visible spectrum. That's an ongoing question, I think. I don't know if any of the panel want to contribute on that one, but it's tricky.
I remember one paper that they were looking more into the sphere part to get some differences. So not exactly from the visible, but they found some differences looking at the sphere bands.
Yeah,
part of the spectrum, but that's the one I come to my mind at the moment.
Yeah, because I think part of the problem is obviously plastics don't have a let's say fixed pigment. They are created in all colors, and also it depends on whether they're floating or on the surface or subsurface. Will have a strong impact on how they can be detected. So yeah, there is ongoing work looking at detection of plastics. Okay, we answered those ones. Okay, so there's a question around if there is a reflectance contribution from zooplankton and whether or not that can be detected. I know there have been some studies, I think, in the North Sea and Barents Sea, where they were associating anomalies, or there were signals associated to chlorophyll that may actually be from large zooplankton blooms. I think that was maybe from, I'm trying to remember if that was from McKee. I could look it up, but yeah, there is, if you get really, really dense blooms of zooplankton, they can technically be visible.
Tom, if
yes?
we are thinking about the same paper by Dave McKee was involved, but that was in the high latitudes, and it was specific for Calanus finmarchicus, which has a pink color. So they were able to show that if these pink colored zooplankton are present in sufficient numbers, then you could distinguish them from other things. But I'm not aware of a general algorithm for detection of zooplankton. Many of these questions about can we do this, can we do that, you will be able to answer them yourself if you follow what Anna was telling you. The ocean color is an instrument that detects changes in water leaving signal in the visible part of the electromagnetic spectrum. So if you want to think about whether satellite can see something or the other, try to think about what optical signature it might have. What is the magnitude of that signature? Is it close to the surface? And are you likely to see it from space? Lauren in her lecture gave this very simple method. She said, look at the water using your eyes. Eyes are very similar to these satellite detectors, except they are up in space. So if you can see them and distinguish them, and if they are present in sufficiently large size, then the chances are that the satellite will pick it up. But many things that your eyes can't see, it would also be difficult for the satellite to see. But this is only a rule of thumb. There are exceptions. Phytoplankton, if you look at them in the very low concentrations in the water, you can't see them because they are minute, they are low concentration, and you miss them. But if you pull back, those small signals become sufficiently integrated that you can detect them. So these are the things you have to think about. Now, if something has no optical signal that would allow you to distinguish from other things, or if it has no optical signal at all, then you can't detect them. One example might be nutrients. Nitrogen absorbs in the UV, and where do they absorb? Some of them have an absorption in the UV part of the spectrum, but they don't absorb in the visible part, so you can't detect nutrients from space. So that's the general rule of thumb. Think about their optical properties. Think about whether they have a clear signal that you can use it to distinguish from other things, which might appear as a background. And if there is, then you can go developing an algorithm to detect them.
And I think this slide is very, let's say, promising for the future, because again, one of the problems that has been addressed by a number of those points is not only can you see it, but can you distinguish it from something else that might contribute? And on the top here, we've got Prisma, and in the future we will have PACE, and CHIME is also a hyperspectral satellite coming up. That's more for land, but you could use it in the coastal zone. And those are hyperspectral, which means you have more degrees of freedom, and you may be able to separate some of the components where before we couldn't, because you will have independent or more independent measurements across the spectrum. That's not true for everything. Some things still will always look the same or similar, but we might have a little more information with which to tease apart some of these signals. So the future is interesting, bright.
And if I may add something, for example, another example like nutrients is salinity. But actually another strategy that you can find, I mean, because you can find papers saying, okay, salinity from ocean color, but actually another way or strategy to try to estimate things that don't have the spectral optical characteristic that can be detected. But if locally it is correlated to another substance or another group that changes the color, you can get your local algorithm to estimate chlorophyll. For example, sediments, it has been done with rivers, which is highly correlated, the amount of sediments with salinity. So there's like a walk around to use this data, but basically it's local. If you don't have this correlation, you will not be able. It's not that ocean color can see salinity, but using ocean color and its correlation with another or nutrients, as also Shuba said, it can be used.
Yeah.
It cannot be directly derived, but it can be used if in your local region it's highly correlated to another variable you're interested in.
Good point, Anna. Yeah, if you can, if you can't detect it directly, you can try to think of a proxy. And if the proxy is detectable because it is correlated with what you want to detect, you can develop some indirect algorithms to map them, even though the satellite is not directly seeing them. Tom, there is a question here I see about algorithms failing in turbid inland waters. Could I try and answer a little bit about that? And I'm sure Anna will want to add to that anyway. So the question is that I'll read it out. Atmospheric correction for turbid inland waters is very complicated. What is the solution if we already applied several atmospheric correction methods, but the result is not satisfactory? It is a difficult thing if you are in a really optically complex waters, such that standard algorithms, even those developed for coastal waters, fail. There is no easy fix to such a situation. In our case, and I can only give you a specific example. In our case, when we were working with a complex inland coastal water body, we also found that the general algorithms were not satisfactory. The only way we could work around it was by carrying out some field experiments, field campaigns. We collected in situ data, and then we tried to empirically correct the acolyte and what was the other one? Polymer algorithms using the in situ data, and then applied the correction. I'll put the reference in the chat, but there might be other ways of doing this. But when it gets really complicated, you really have to resort to in situ observations and try to match the algorithm for the specific case that you are looking for, I think.
I see a question. Oh, sorry, go on.
May I add something?
Yes, please.
Apart from the in situ measurements that you mentioned, I think now, for example, with the development of artificial intelligence and having, for example, the hyperspectral satellites coming, there may be also a chance to tackle more complex problems. Right? So I think that's also, if we look into the future, that's another way of getting into, again, more complex waters.
But also having in situ data of your site, this artificial intelligence will do if you give good data. So I think also what should we mention, the optical reflectance, the characteristic of your water will be necessary also for these algorithms to work properly in your place. But yes, yeah, you're right.
Since my student days, I have this dream that we will have a sort of a cascade of algorithms, of branching algorithms. And as you go towards each specific case, then an algorithm tailored for those conditions will kick in. But we are not quite there. But these artificial intelligence kind of methods may take us towards that. And as Tom also mentioned, as we go towards hyperspectral, that will also help us address some of these really naughty problems.
There is a question around the provision of satellite data. So we have a whole lecture coming up on data access, but the question boils down to high resolution imagery being available only in limited locations from providers like Platt. And there's a question about how much is provided by ESA or NASA. I think my quick answer would be it depends on what you mean by high resolution. So ESA provides, so the Sentinel-3 data is global. We have 1 kilometer and 300 meter data available from Olchi. That is 300 meters in the ocean, is deep ocean, is very high resolution because things don't vary over those spatial scales very much. But yes, when you get into the coastline, you might want tens of meters of resolution. And that's where you might start to look at data from something like Sentinel-2 or other sensors. But as far as I know, ESA and NASA, it's not about the processing effort required. It's more about that the satellites are tailored to specific resolutions, and it isn't worth, measuring 10-meter resolution across the deep ocean because it just doesn't vary at those scales. But I don't know if the panel wants to add anything else to that. But it's whereas Planet might say that it's a processing computation cost, the space agencies tend to do everything on bulk, but the limitations will be at the mission level because it was, the satellite is designed for a certain purpose.
In general, we can say that Sentinel-2 does collect data in coastal areas along with the land. So does Landsat. and that is we use them quite a bit for very coastal or inland water work.
Yeah, because originally they were thought as land
That's right.
missions, but we're using the coastal, which has land, but also the waters. So
That's right. Yeah, as long as there is
some land.
some land attached to it, we can get the water data.
Indeed.
Okay, we're getting towards the end of the questions, I think. So there was a question again coming back to if measurements containing seabed irradiance are marked as contaminated, does it mean that whenever you see the seabed, you get an invalid product? It's my quick response, again, others can contribute to that, would be that when I said, or when we said that if you detect the seabed, you might mask it, that's again for these large default processing systems, because they're designed for more open ocean. As you move into coastal waters and you start to get more tailored algorithms, in fact, as we've already discussed, you might be interested in trying to map what's on the seabed. Obviously, you don't want to mask those pixels. So it's more about making sure that when you set up your processing chain, you have the correct flags. And I'll go into this a little bit when I talk about the climate data processing, about all the various stages and things you want to check you have or have not turned on or off when you build a standard ocean color processing chain. Yeah, so you can measure or estimate chlorophyll A in shallow waters, but you should be aware that with some of the let's say flags or safety measures that you turned off, your uncertainties might be higher. Yeah.
Tom, may I come in with a general comment?
Yes.
I wanted to mention to the participants, if they are not aware of it already, that there is an organization called the IOCCG. It stands for International Ocean Color Coordination Group. It is also one of the sponsors of this training course. The reason I mention it is that they do a tremendous job of producing high quality reports. They're actually at the level of monographs on the topics on different aspects of ocean color technology and applications. So if you are not familiar with them, this is a wonderful resource that you can use to when you are on your own and you have all these questions and there's no expert in house to answer them. So please do, I've just posted it in the chat. Please have a look at them, and you will see that there are, I don't know, I think there are over 20 reports now that address various aspects of ocean color technology, the science, the applications. And you may find that many of the questions you have asked here are answered in depth in one or the other of these publications. So as a future resource material, please do have a look at that. If you write to them, they also send you a hard copy, but all the reports are available online as PDF documents.
Okay. I think we might have time for maybe one or two more questions. There's one that is a quick one, which was around sometimes you see this foam when you go to the beach and it's all been wavy and stormy, and is that CDOM? My understanding is that's Phaeocystis. but I don't know if Anna would like to, or anyone else would like to expand a bit more on that. That's
I think it depends. Sorry, I'm not
Okay.
really, I don't live close to the sea, but it happened here in Argentina. So what's that? But sometimes, yeah, actually it's more of the breaking waves. So they break the sails. So it's more kind of, and
I mean, it's definitely not what you would call dissolved organic matter because it's not
no,
dissolved in the water, right? This is
no.
a surface floating foam. Usually around the UK, at least, it comes from an organism. Yeah, so the waves basically aggravate and disturb the algae, and there's a particular one called Phaeocystis, which makes extracellular kind of mucus, and this ends up causing the kind of foam or bubbles that you can find at least around the kind of mid-latitude or northern latitude waters.
Indeed. Yeah, but definitely it's not dissolved, it's just surface.
Yeah, it's not dissolved. Yeah. And there was a question around chlorophyll A as a product and how that does that cover also other chlorophylls, because we have chlorophyll B, chlorophyll C within this list of pigments. So I was just wondering if you could expand a little on the difference between the chlorophyll pigments and what we're actually detecting when we use these algorithms.
Yeah, well here it's I'm showing again this with actually we're using the it sees the total absorption, so the sum of all the pigments optical characteristics. So basically using the blue where the chlorophyll A dominates, it doesn't mean we're only estimating chlorophyll, but actually is the sum, but in the blue it is mainly related to chlorophyll A. For distinguishing other type of chlorophylls, you need specific bands, and also I know you have to you might do it, and I saw a recent paper in the southern North Sea when you have a specific phytoplankton species that has this so it's a bloom, so in large quantity, and it's a specific of this species that has a high amount of chlorophyll C, let's say, I don't remember which one, and you have, for example, the work that has been done to differentiate them was with a hyperspectral. So actually what they did was like a time series of chlorophyll, and they know they have this, I think it was Phaeocystis blooming in the spring, but also they have this chlorophyll peak in the winter, and they know they don't have that. So actually when they did this second derivative of the reflectance and found this slight difference between the chlorophyll A and C, so they can detect it in that specific condition. So it's a blooming that there's chlorophyll C, and that, but actually with chlorophyll, this type of algorithm, it's not possible to differentiate. It's just total chlorophyll or phytoplankton, yeah, chlorophyll A, basically because of the peak of the absorption of the chlorophyll A.
Okay, I think that's most of the questions. If I've missed any, I'm sorry. There were a few questions that definitely came up that I think will be answered more fully in future lectures, be those around data access, derived products, processing schemes, and tools for applying your own atmospheric correction, for example. Yeah, so I think we'll probably draw it to a close there. Thank you all for attending. Thanks to Anna again for a fascinating lecture.
I was just putting in a chat, somebody was asking for references. Uma Maheshwar Rao, for references on sea grass mapping, and coral mapping, I think. And I was just putting it in the chat that I don't work on that area myself, but after I saw the question, I went on Google and searched ocean color and seagrass, and quite a few references came up. So if I wanted to work in that area, that is where I would start. I know that in fact, we have one of our own projects that also has a seagrass mapping component,
Yes.
but
Yeah,
that
that's
part is led by Tom. Can you put the university reference or link there?
yeah, that's DLR. I'll send that, put that in. So find. For example, so this is a paper from Dimos Flaganos, which is looking at seagrass extent mapping across the Mediterranean. But that group does a lot of work on seagrass, the seagrass beds. Okay. So yeah, I think that's the end of this lecture. So thank you all. See you again next week. So next week is the lecture on data access, I believe. So we'll have a talk from Dr. Ben Loveday, who will be covering all sorts of ways to get hold of all the data so that you can start doing all of the things that we've seen, started to see applications of, and you can start experimenting yourself. So thank you very much.
Thank you, everyone. Above all, thank you very much, Anna.
Thank you.
That was lovely. Thank you very much.